Good evening. My name's James Danger. In this video, I was talking about the oviductal extracellular vesicles, EV or EVS, and its impact on embryo development on a cold Australian winter's evening. So this article, which appeared in scientific reports from some Chinese authors, published about a week ago, the titled Supplementation with Oviductal Extracellular Vesicles from the Estrus, Meta-estrus, and Diestrus stages improved developmental competency of IVF mouse embryos. So this first part of this talk is all on mouse embryos, and then it's a little bit later on human embryos, and then some of my comments right at the very end. So by way of background, as we know, the oviduct is the place where early reproductive and critical events occur, including fertilization and early embryo development. Recent studies have indicated that EVs within the oviduct can act as mediators of cell-to-cell communications and play a role in mediating these events. However, during in vitro embryo culture, the embryo oviduct crosstalk mediated by extracellular vesicles is absent, which may result in a significant gap between the advanced development of in vivo embryos and the poor development of embryos cultured in vitro. So this is what the authors did. I'm going to try and make it a very quick video. I'm going to try to stop talking and have a shorter video. And so basically the authors collected extracellular vesicles from the oviduct of mouse in the pro-estrus, normal estrus, meta-estrus, and diestrus, or diestrus, meta-estrus. And they produced various concentrations of those, and they added it to mouse embryos either from the time of collection, fertilization, right through to day 4, which in the mouse is the blastocyst, whereas humans stay it's 5 days. And then they added it after the pronuclear stage, the cleavage stage, and the blastocyst stage. And they did one where they renewed it halfway through, and then they had a control. So just some very quick data. That's what I'm going to show you. Basically, in the mouse, if you grow the embryos from day 1, the 2-cell, about 85% will go through to 8-cell stage, 82% will form a morula, 60% or so will form a blastocyst, and about 50% will hatch. But if you grow them in the presence of extracellular vesicles, look what happens. Not only are there significantly more blastocysts appearing, despite even when you added it right from the whole lot, or even just on the last day, look how more embryos you have that are hatching compared to this. It's almost 50% increase in the number of embryos that are hatching. In other words, it's not really affecting the early cleavage stage, but even though this occurs in the oviduct, but the impact is on blastocysts and hatch blastocysts in vitro. The same thing applies if you add it from day 3 or just even on the morula stage just overnight, you still get the same impact. In other words, adding extracellular vesicles to the in vitro culture mouse embryos, right, produces slightly more blastocysts, not an awful lot, but it's significantly more. But really, what's really happening is more and more of those are hatching spontaneously. And just a nice little graph just to show you this is from sets of data for the pro-estrus, the estrus stage, the meta-estrus, and then the diestrus stage. And you can see when they're all the same, but basically they all look the same. This is where you added it on day 1, day 2, day 3, or day 4 at different concentrations. And except for the pro-estrus stage, the other effects are the same. That is, you get significantly more blastocysts and significantly more hatching from both the estrus, the meta-estrus, and the diestrus stage of the cycle in the mouse. And look, you can look significantly, you can see how much more hatching is occurring. But it looks to me like it's largely dose independent, I should say. So just to summarize that, the authors have demonstrated that in the presence of extracellular vesicles, you get significantly more blastocysts and hatching blastocysts. And there's just some suggestion that if you replace it halfway through, that is, they're being absorbed and internalized in the embryo, so they're being used up. So if you add fresh extracellular vesicles, that's what happened in the fallopian tube where there would always be being produced, you seem to get slightly more. So in other words, this kind of either tells you that the vesicles are degrading or they're being absorbed and used by the embryo, and replenishing it with fresh EVs gives you even more hatching than before. So to test the hypothesis, they cultured the embryos in vitro with different concentrations of oviductal EVs derived from the 4 stages of the extracycle in the mouse. The results obtained in the present study demonstrate that the addition of oviductal EVs from estrus, metaestrus, and diestrus stage enhanced in vitro development in the hatched blasters stage. And they make a point that hatching from the zona pellucida is a crucial element in embryo development. If the blastocyst does not hatch in time, successful pregnancy will not occur. And really interesting, if you look at all the work done with euploid embryos in human, the best predictor of success is not a hatched blastocyst, but a day 5 hatching blastocyst. So not only do you have to get a blastocyst, but once they're hatching already, give you the highest pregnancy rate. So there's something here which I think is quite important. It has been suggested that the total cell number in blastocysts was an indicator of embryo viability, and the increase in cell numbers of blastocysts could contribute to hatch from an intact zona pellucida. I haven't shown you that data. And in this study, the addition of 1.2 by 10 to the 10, that's an awful lot of vesicles or particles from the metaestrus that result in a higher total cell number. In other words, you need a lot of EVs to get you more cell division occurring in the embryos. Also, addition of extracellular vesicles from the diestrus stage significantly enhance the total cell numbers regardless of additive doses. Moreover, the results show that the effective addition of EVs from the proestrus stage is lowest, and oviductal EVs from the diestrus stage had the best effect on blastocyst hatching and cell numbers in the mouse. So in vivo, extracellular vesicles released by the oviductal epithelium are internalized by target cells in the embryo and degraded, and the newly produced EVs are continuously resupplied. However, in vitro culture systems, as EVs cannot be replenished in a timely manner, the balance between EV generation and degradation has been disrupted. The lack of EV regeneration in vitro culture systems may increase the risks of oxidative stress, autotrophy, and apoptosis, which is likely to impair embryonic development. Overall, in this study, the results have shown that the addition of oviductal EVs isolated from various stages of the estrous cycle in the mouse to culture media can benefit embryonic development in vitro, which is consistent with previous literature, which I haven't bothered to review. So far, EVs have not been truly tested in human-assisted reproductive technology, nor have they been widely applied in the animal reproduction and breeding. However, what's really interesting was when I was making this video, what happened, but this paper appeared in the Journal of Extracellular Vesicles. Never heard of it. I will never ever found it. It's not part of my scope, but just goes to show some of these papers appear in the most weird journals out. You would never have predicted to look for it, but this is kind of an interesting one and supports what I've just been talking about. This is also from Chinese authors. And where they say the extracellular vesicles from human fallopian tubes enhance IVF embryo development and contain functional proteins, including, excuse me, YHH, YWHAZ, whatever that is. I don't know what that is. I'm not a molecular biologist, but I think we're all, as embryologists, we're all going to learn very quickly what all the different proteins are in these extracellular vesicles. So I just... The extracellular vesicles in the mammalian oviduct constitute a key maternal regulatory system that maintains redox balance during early embryogenesis. Yet their molecular cargo and functional relevance in human embryos remains poorly defined. Here, we show that human fallopian tube-derived EVs are rapidly internalized by human pre-implantation embryos and improve development quality in vitro, increasing high-quality day 3 embryo formation and blastocyst development. Now, I'm only going to show you just a little bit of data from this paper, and I think I'll come back and do a separate video on it later on. There's an awful lot of information, and it contains both human and mouse work. And it's actually, even for most embryologists, including me, I'm a bit of an old fuddy-duddy, it's actually quite outside my scope of experience. But I sense within this something that's very important, something that we don't see in human IVF, that in fact the embryo doesn't exist by itself. And I think when I do that next video on this thing, I'm going to do a couple of slides on the old aspect of group culture, where they've often thought that each embryo, good embryos help other embryos develop, but it may well be through a system of embryo-derived extracellular vesicles. But this one is from human fallopian tubes. And basically, the zygotes were derived from discarded immature oocytes that were cultured in IVM and fertilized when they became mature with the husband's sperm, but they were never transferred. They were cultured at the blastocyst stage and then discarded after growing them with the EVs from other women. There is other work in this paper with mouse embryos, and they swap and change between the data, so you have to keep your wits about you. And this is the location of the article, and I recommend everyone should read it. I'm really going to show you one set of data from it. This is all the mouse work, and I don't really want to get involved at this stage. There's a lot of molecular biology, and I have to admit, I don't really understand the molecular biology of the proteins found in extracellular vesicles at the moment. But so here they can show that the content of extracellular vesicles are being taken up by mouse embryos, right? And they reduce DNA damage and Ross levels, etc. But the top line here is human embryos. So these are grown from immature oocytes at collection, cultured by in vitro maturation until they produce a hollow body, inseminated by ICSI and grown to blastocysts. And so the development rates are quite poor because they come from really quite immature oocytes. But look at this. So it made no difference in the percent of embryos that developed into day 3 embryos, cleavage rate. But look at the number that high quality. Instead of having about 5 out of 48, there were 9 out of 30, right? Going from 10% to about 35% high quality human embryos. The blastulation rate 7 out of 48 in the control and 11 out of 33. So the numbers aren't great. I don't need to. I mean, it may well be this just doesn't stand the test of being a bigger study, but I just thought it's really interesting. So even from these really poor embryos, these immature oocytes, you get a few blastocysts growing. You almost double or triple the number of blastocysts forming just in the presence of a human fallopian tube extracellular vesicles. And I thought this is a really interesting one. That is, if you look at the degree of fragmentation, most of these embryos that are grown in the presence of extracellular vesicles have almost no fragmentation at all. So I think I'll come back and do another talk on this in a month or so. But I thought I just want to tantalize you saying that there is data now, albeit on immature oocytes, albeit with only few number of embryos, right? And so it doesn't mean a great deal, but it's the start of something which I think is really important and could potentially transform human IVF, taking us from a very low pregnancy rate to potentially even doubling it, because all the data suggests it improves the hatching rate. Okay, so the YWHAZ protein is also a 14.3. I don't even know that terminology. But what I can say is that it's a highly conserved cytosolic adapter protein that regulates client protein localization, stability, activity, cell-to-cell progression, apoptosis, and stress response. In other words, it stops an embryo from undergoing stress, which we know is the biggest cause of aneuploidy and delayed development. They found that YWHAZ is highly expressed in human day 3 embryos, but it declines in blastocysts, suggesting a temporal requirement during cleavage stage. In other words, it keeps the embryo going for the first couple of days, but it runs out of puff, but it needs to have input from the oviductal environment to keep going. It's a bit like a test. Immunofluorescence reveals cytoplasmic localization of YWHAZ in human embryos, which is consistent with its role as a cytoplasmic signaling adapter. So I don't need to say anything more about it. Is that there are a whole lot of different proteins found in all these different vesicles, and they're just picking on one. But this was the one that was it was absorbed by or taken up by human embryos more than any other protein. Together, these findings support a model in which YWHAZ contributes to redox homeostasis in pre-eclampsia embryos and requires vesicle-mediated transport for effective delivery. It's interesting that these vesicles can go through the zona pellucida, which of course is porous. I think there's something really going on here. This paper also describes some synthetic EVs, and when I do that paper, if you want to go and read the paper yourself, you can see where it's going. Then in the future, you may buy media from a supply company, but you buy a whole, you might modules of EVs containing different proteins that you add to the embryo on day 3 or day 5. And how that manages, I have no idea, but you can kind of see where it's going. From a translation perspective, defined EVs, derived factors, or engineered EVs, that's what I was talking about, based delivery systems may provide a future strategy for improving IVF culture media, particularly by restoring redox regulatory signals that are insufficiently supplied by current in vitro culture systems. This could be a landmark paper, or it could be just a load of rubbish, and time will tell, but I suspect it could be a landmark paper. So my comments are this: right from the first in vitro culture, embryos have been grown in cell-free media. Although when I first started mouse IVF back in the late 70s, we often grew our embryos in the presence of cumulus cells, and they always grew really, really well. In fact, some of my best embryos I've grown in the mouse were the ones where we grew them in the presence of all the cumulus cells that were left over from fertilization. It's not quite the same. In other words, it's some sort of conditioned media. In fact, they are able to develop and implant as the generations of embryologists to consider the embryos as self-sufficient. In a way, this is correct. We don't need anything else, just simple media with a few bits and pieces, right? And the embryo is quite capable of doing it itself. This is this quiet embryo hypothesis of Henry Leese, etc. But the disappointing pregnancy rate, even with the best euploid embryos that we can create, tell us there are many other factors operating out of sight. Now, I did some work back in the 1980s with PAF and mouse embryos, so I already have a predisposition to believe this effect. But reality is people have been working hard for the last 20 years to get the pregnancy rate up, and no one can get above 60%. But in reality, most pregnancy rates are down around about 30-something%. Extracellular vesicle positive effects on embryo development remind us that there remains a lot of in vivo influences that the in vitro embryo is denied access to. Like hydroponically grown fruit, it works, but tastes different to that nurtured in your own backyard garden bed. There are several EV sources that may influence the ability of embryos to hatch and the uterine epithelium to receive that embryo. And I don't think we've even scratched the surface of it, but I think you need to mark for your bookmarks anything about EVs in the future. Maybe we're moving into a new phase where we release life. Life is far more complicated than a sterile, lonely world of IVF. So my name's James Stanger, and I'm an embryologist with more than 50 years experience. These videos are part of my Third Age YouTube channel in an attempt to bridge the gap between information you may need for your fertility treatment and the most recent publications available to the staff. I'm not a medical person, and this information is not medical advice, but it may help you knowing what questions to ask about your treatment with your doctor, and maybe to help in understanding the advice you receive. If you like what I'm doing, please subscribe, press like, and leave a comment. Just like to acknowledge that Third Aid is the primary source of the sponsor of the channel, and my Third Aid EQA program, which is another one of my activities, is an online EQA and training program in IVF, andrology, and embryology. So I'd like to thank you if you persevered for so long. Still 19-minute video, still far too long. IVF is a personal journey that involves the action of many different concepts, ideas, starting techniques, and helps to understand as many aspects of that journey as you can. I hope this information may be of value. If you like the information provided, please tell other IVF clients about this service, and please subscribe. And at that point in time, I think I'll say goodbye and good night.